Thank you very much for attending our webinar called Nano-Emulsions for CNS Delivery of Biological Therapeutics. I'm Stephen Masidi. I work for Microfluidics, and with me is Dr. Mansoor Amiji. I'm going to talk for about 5 minutes, and then I'm going to put Dr. Amiji on the line, and he's going to provide the bulk of the presentation. Afterwards, hopefully we'll have about 5 or 10 minutes for questions and answers. So if at any time you have any questions, please feel free to send us a message in the Q&A tab at the top of your screen, and we will try our best to answer it at the end of the presentation. Or if you prefer, we can answer your questions afterwards by email. You have my contact information, and I'd be more than happy to answer any questions. If you just send me an email after the presentation. This is just a quick slide that talks about microfluidics. We've been in business for 32 years. We have many of thousands of customers worldwide. Microfluidizers produce nanomaterials for many different applications, primarily pharmaceutical ingredients, and there are several drugs that have been approved for release and delivery that are currently on the market. This is basically what we do best. We create nano-emulsions. We do cell disruption for biotechnology applications with very high protein recovery. We do very efficient particle size reduction and encapsulation, and we work with both nanodispersions and on deagglomeration applications. This is the basic schematic of a microfluidizer processor. Essentially, there's an inlet reservoir which enables for a continuous process of emulsions or suspensions. It can accommodate materials with high solid loading and also high viscosities and also very high temperature ranges. One of the key characteristics of the microfluidizer is the constant pressure intensifier pump. That's pretty important because it delivers all of your material at a very uniform processing condition to the interaction chamber. The interaction chamber is really the heart of the microfluidizer processor. It is a fixed geometry path which ensures that all of your material is processed at a very consistent shear rate. This is a quick cartoon of a couple of different types of interaction chambers. The one on the left is the Y-type interaction chamber. This is generally used for emulsions and liposomes and mixing applications. The chamber on the right is the Z-type interaction chamber, typically used for particle size reduction, dispersion, cell disruption, deagglomeration. They're both made out of extremely resistant, wear-resistant materials like polycrystalline diamond, and the other feature is they're very scalable. The main benefit of the microfluidizer processor is the extremely high shear rates that we are able to achieve, much higher than any other technology, and also the inherent scalability of our process. We are able to place microchannels in parallel when we scale up to ensure that the processing conditions that you were able to achieve on your lab scale will be able to be replicated exactly on the production scale. This is just a quick summary of what I just said. These are the key features and benefits. Our technology is a constant pressure process with extremely high potential processing pressures up to 30,000 psi. We use fixed geometry interaction chambers and multi-slotted interaction chambers. This results in very small particle size with very consistent processing conditions, resulting in very narrow particle size distributions. And also, we guarantee scale up from lab scale to production scale. So on the right there, you see there's the LV1, which is capable of processing 1 milliliter of material. And then we have a biopharmaceutical 725020, which would be able to generate the exact same results in CGMP environment at 8 liters per minute. So without further ado, I'm going to pass it over to Dr. Amiji. He is going to be able to take us through his presentation.
Okay, good morning, everyone. Thank you, Steve, for the opportunity to present, and I hope everyone can hear me. So today I'll be talking to you about our work here at Northeastern University in Boston, specifically on development of nano-emulsion-based delivery systems to help in carrying drug molecules into the brain. And more importantly, we are interested in the class of molecules called biological therapeutics. So just to give a brief background on biological therapeutics, one of the challenges in the 21st century right now are the diseases of the brain, especially those that are chronically affecting some of the elderly populations, such as neurodegenerative disease. The numbers are staggering. The current trend projects that about 14% of the population in the world will have age-related neurodegenerative diseases by the year 2020. And this especially is troublesome because in many countries around the world there's an aging population. The life expectancy is increased, and more importantly, some of the challenges with development of therapeutics are quite astounding. And one of the interesting thing about the molecules that we and others are interested in looking for therapeutics in the brain are these biological therapeutics, and they include peptides, proteins, nucleic acid-based molecules such as small interfering RNA, as well as plasmid DNA, and then also in some cases whole cells, either modified cells or even cells such as stem cell transplants. And those are all considered to be biological therapeutics because they have the propensity to modify the disease outcomes, not just treat the symptoms. And I'll explain some of that in the latter with some specific examples. The problem, as many of us know, is that how do you get these molecules into the brain is very challenging, and it really creates a huge problem in terms of development. This work was first highlighted in an article in 2010, and you see in the bottom part of the slide that CNS diseases or drugs that are developed for CNS diseases actually take the longest time to get to approval. There's also the success rate for these types of molecules is lower compared to some of the other disease areas, and the cost is incredibly high, which means that a lot of companies that have programs in CNS therapeutics are rethinking or have basically withdrawn from development of CNS therapeutics because of some of the challenges in getting the drug into the brain. So we and others have thought about ways to get these drugs, and in the next slide what I show you are some of the barriers from getting drugs from the bloodstream into the brain. One of the biggest of these is called the blood-brain barrier. And many individuals think of the blood-brain barrier as simply a physical barrier, meaning that the cells of the capillary endothelium are lined in such a way that they create these tight junctions and prevent drug from getting from the blood side to the brain side. And these tight junctions basically is almost like a a wall that prevents the drug from crossing over. But in addition to just being this tight junction, these cells also express various types of molecules, and it's actually the biological barrier to drug transport is much more profound than the physical barrier based on the tight junction. The biological barrier comes from having these molecules on the cell surface. Some of these are transporters that take the drug from inside the cell and push it outside. These are called efflux transporters, and one common example of this is a molecule called P-glycoprotein or PGP. The other biological barrier, the blood-brain barrier, are enzymes that basically degrade molecules, and biological therapeutics, especially peptides, proteins, and nucleic acids, are highly susceptible to degradation by enzymes. So crossing these molecules across the endothelium and into the brain becomes incredibly challenging. The other problem with delivery into the brain, although it's not as much appreciated, is the fact that in order to get from the blood to the brain, one also has to think about the distribution, metabolism, and clearance of the drug in the systemic circulation itself. So when you inject or when you take a product orally, the drug gets into the bloodstream, and then from the bloodstream it has to actually traverse and go across the brain. But if the drug is rapidly diluted in the bloodstream or is rapidly metabolized and cleared, then the ability to cross into the brain becomes even more difficult. And even for small molecules, that's a challenge, because you can have a very hydrophobic molecule which has a very high volume of distribution in the systemic circulation and doesn't really get an opportunity to cross over into the brain. But this is a much bigger problem for biological therapeutics, because they are hydrophilic, they tend to be very labile, and subsequently are very, very susceptible to dilution, metabolism, and excretion from the body. So how do you make these molecules cross into the bloodstream? And so the approaches that people have tried involve two major categories. You either can do invasive administration, or what we are interested in is the non-invasive methods. Invasive method means you basically have to create a catheter that delivers the drug in the brain. For example, ICV is an example of a catheter that is placed through the skull and directly delivers to different regions of the brain. You can disrupt the blood-brain barrier by an osmotic mechanism, for example, using mannitol, which temporarily opens the tight junctions and allows molecules to go through. But these openings is transient, but at the same time, it's difficult to estimate how long does one need that tight junction to be open, and what else can cross over. Delivering intrathecally and then having the drug go into the brain through the cerebrospinal fluid is another mechanism. But we are actually interested in the latter two that I've highlighted at the bottom. How do you use nanotechnology in delivering, especially engineering nanotechnology, to enhance drug delivery from the blood to the brain? And then on this last example, I'll talk about our approach to use intranasal route, basically delivering the drug from nose to brain, which also is a very important mechanism that completely bypasses the blood-brain barrier. So I'll approach this seminar or this webinar through these two different examples of using nanotechnology approach to get the drug from the blood side to the brain. So this is when you inject the drug or when something is taken orally. And then the second approach is a non-invasive mechanism by which we can deliver through the intranasal route and get the drug absorbed through the olfactory mucosa and all the way traversing into the brain, and show you some examples and work that we have done in this area. So let's start with our first example, and I had a PhD student, Lipa Shah, in my lab who worked on this project, and we use nano-emulsions. Nano-emulsions are basically oil and water emulsions that are made specifically in compositions that allow us to make them in a very small size range. And I'll give you details of this as we go through this slide. We are specifically interested in using edible, safe oils that are rich in omega-3 polyunsaturated fatty acids. So in this example, we have used fish oil, which has a very high concentration of EPA and DHA, the two common omega-3 fatty acids that are found in fish oil. And then flaxseed oil, which is also very rich in omega-3 fatty acid that comes from plant seeds, in this case, alpha-linolenic acid. And if you see in this table, we make emulsions using two different approaches. The first approach that you see up here is designing the emulsions using an ultrasound approach in the lab, which can generate small volumes up to about 5 ml to 10 ml. So for lab scale, we use this ultrasound approach, basically using an ultrasound sonicator, and we can reduce the oil particle size down to a level of between 100 to up to 300 nanometer, depending on formulation. The second component, so there are three components in an emulsion. It's a very versatile system. The first is obviously the oil, the selection of the oil. And for us, we think about the oil for two purposes. One is where we want the drug to go. In this case, when you want to go to the brain, we select these omega-3 rich edible oils. And then the second component is the surface modifier to make sure that the oil droplets remain stable when they are in the emulsion form. And so for our purposes, we have used number of different kinds of surface modifiers. We actually combine different kinds of surface modifier to make sure that we get a stable formulation at the end. We will use phospholipid-based surface modifiers such as lipoid 80 or cationic surface modifiers such as sterilamine. And then we also use polyethylene glycol modified phospholipids such as DSP PEG or tween 80 or pluronic F65. And the choice of the surface modifier, the composition, the concentration of surface modifier is critical in order to ensure stability. And one of the most common questions that I get when I give these kind of seminars or webinars is how to ensure that the oil droplets will not coalesce. And this is really why, it's really critical to select the right kind of surface modifiers or emulsifiers that allow us to make a stable formulation. And we test all of our emulsions both at 4 degrees Celsius as well as at room temperature. And we ensure that even for lab scale preparation methods, we have stability for up to 6 months. And we will measure that by using light scattering approach. We'll measure that by using to make sure that in the oil and the emulsion remains stable. There is no phase separation between in oil and water phases. And we want to make also sure that the drug that's encapsulated remains stable. The second approach, which is what I'm really interested in talking about, is if you want to scale this up, we are using the Microfluidics Microfluidizer, the unit that you see at the bottom there, which has been donated to us by Microfluidics, and we continue to use this for making different kinds of nanoparticles. Today I'm going to talk about just our work with Nano-Emulsion. And at the right-hand side, you see the emulsion that we make. This is the TAM image. But using the microfluidizer, we can make up to 15 ml in larger volumes. We can go to liter quantity very, very easily. As Steve mentioned, the instrument has the capacity to generate very rapid process and allows us to control the size and the properties. And what you see in the table that follows is our working formulation, which is made with fish oil, has lipoid 80, which is the phospholipid phosphatidylcholine, and then we add a co-surfactant, which is tween 80 in our case, which also has the pegylation. It's pegylated, but more importantly also allows us to get these emulsion droplets across the blood-brain barrier. You also see the size, the particle size with the microfluidizer using about 16,000 psi up to 20,000 psi. We are able to generate it under 100 nanometer size range. So the oil droplet size range gets to be below 100 nanometer, and we can reproduce that. You see the polydispersity index is very low, and the surface charge on the oil droplets, the zeta potential value, is negative because of the presence of phospholipid and tween. If we use other kinds of co-surfactants such as sterilamine or DOTAP, we can generate this positive surface charge as well. We can also use other type of co-surfactants such as bile acids or bile salts, and that helps us to increase absorption across the GI tract if you want to design something that's orally bioavailable. So there is a tremendous amount of versatility in the design of these nano-emulsions, both for oral absorption, but more importantly for this webinar, CNS delivery. So first, we worked with Professor Rebecca Carrier, who is our chemical engineering faculty here at Northeastern, to test if our nano-emulsion system can actually enhance permeation through a model, an in vitro model of the blood-brain barrier. And we use the Transwell system, which you see on the bottom left-hand side, to create a cultured a cell culture system where we can look at permeability from the apical side, which is the top side, to the bottom side, which is the basolateral side. And when the cells are grown on these cover slips that are put in the Transwell, you can actually have very good polarizations, which means that these efflux transporters are going to be expressed on the apical side, but not on the basolateral side. And so you can look at transport from A to B, and then again from B to A, to characterize not only the tight junction transport, but also the efflux that occurs due to this expression of various types of efflux transporters. And we characterized, so we tested a number of different cell systems, and we found that, interestingly, the Keiko2 cell, which is actually an intestinal model cell of for intestinal absorption, makes a very nice tight junction and allows us, and also expresses a lot of the efflux transporters that are seen on the blood-brain barrier. So we use this as a model to characterize our emulsion transport as compared to a solution form, just to understand whether we can actually enhance permeability and also availability from apical to basolateral. For these studies, we use a model system. We use the rhodamine 123, which is a fluorescent dye, but it's also well known to be a transport substrate for this P-glycoprotein, which is a protein expressed by the MDR1 gene. And so in the next slide, you see the permeability changes that occur when you put rhodamine 123 in aqueous solution or in our nano-emulsion. The same nano-emulsion I described before, made with fish oil and has this both lipoid 80 as well as tween 80. What you see in this graph on the top panel, the A side, is the solution form of rhodamine 123, and the B is the nano-emulsion form. The green triangles is a ratio of what we call the efflux ratio, which is basically the ratio of the permeability from the basolateral to the apical, which means that if there is high efflux, then the transport is not as efficient. And you can see that the red bars that you see are basically transport that's coming from the basolateral to the apical, where there is no efflux transport, whereas the blue bar are the transport that's coming from top to bottom, where there is efflux transporter. So the less efflux transport, the less transport from A to B means that there is a greater efflux ratio. And that's what you see with solution of rhodamine 123. There's a very high degree of efflux. But if you use nano-emulsion, which is on panel B, you can see that there's a greater transport from A to B, which means that nano-emulsion actually are able to overcome the P-glycoprotein efflux. And the reason for this is because nano-emulsions are endocytosed by the cells, and subsequently the transport occurs paracellularly and avoids the efflux pumps, because the whole oil droplet is able to ferry the molecules across, and you don't need the solution, you don't need the free rhodamine 123, which is what is the substrate for the PGP. So we've been able to lower the efflux ratio tremendously by using the nano-emulsion form. And then we also tested a very interesting idea that could we silence or reduce the expression of these transporters? And curcumin, which is a very common spice in Indian and Asian food, is a well-known inhibitor of different kinds of efflux transporters, and has been well characterized, and we have done a lot of studies with curcumin to enhance absorption from the GI tract. But in this study, we wanted to see if curcumin could actually also inhibit transport of these efflux transporters at the KECO2 cell junction. And what you see is that when you give curcumin pre-treated on these cells, you are able to decrease the expression of efflux transporters. We see the expression of MDR1 decrease with both 5 micromolar as well as 20 micromolar dose of curcumin when given in nano-emulsions. And we also see some other transporters such as MRP1 and BCRP levels reduced tremendously when you give these curcumin pre-treatment at 20 micromolar dose. That's shown in the graph on the right-hand side. But MRP2 is not changed. The levels of MRP2 are not affected by curcumin. So clearly we have been able to reduce MRP1, MDR1, and BCRP genes, which are all very important for basically effluxing out the drug from in the lumen of the arteries or in the lumen of the capillaries in the brain. So with this pre-treatment, we then tried again our permeability studies with using rhodamine 123 both in solution and in nano-emulsion form. And you clearly see that even in solution form, when you pre-treat with curcumin, you are able to show that you're inhibiting these efflux transporters. And now we've been able to suppress the efflux ratio tremendously compared to the previous example when we didn't pre-treat with curcumin. But the effect on the nano-emulsion is even more profound. And here, again, we believe that nano-emulsion not only in some ways helps to get the entire droplet gets across the cell and gets us across, but also there may be some free rhodamine 123 that is released from the nano-emulsion, where, again, with the fact that the efflux transporters have been inhibited, you see this overall cumulative enhancement in permeability across the KCO2 cell layer, which is a mimetic of the blood-brain barrier. So we were really excited to see these results in vitro, and then we started to think about, let's do some in vivo studies to actually see how this relates to an actual therapeutic molecule. So we got very interested in chronic pain management, and specifically thinking about what are the different kinds of therapies that are used in treatment of pain. And as you know, pain is defined, there are a number of different classifications, chronic pain, acute pain. We are mostly concerned with chronic pain. And in chronic pain, there is a nociceptive pain as well as neuropathic pain. The neuropathic pain is one that is most debilitating. This is the kind of pain that occurs in diabetic patients who, at some point, the pain is so severe that even a small stimuli, such as feather touching their finger, would actually exacerbate into a very, very harsh pain sensation. And the current therapy for pain is based on the step-ladder approach, which is characterized from the World Health Organization, that pain is first identified, and then initially you start patients with non-opioid treatment, usually non-steroidal anti-inflammatory drugs or some other analgesic products which are not in the opioid classification. And then as you get to mild to moderate pain, then some of the oral opioids are indicated. And then as you get to severe pain, such as pain that's associated with cancer and cancer metastasizing to the bone, then these patients are put on chronic opioid therapy. So opioids obviously have a huge role to play in treatment of pain, and we got interested in opioids for a number of different reasons. One is the fact that they're very, very potent drugs in treatment of chronic pain, and so could we develop a therapeutics around opioids that would actually be safer and help us to treat pain in a much better way? And so for that, we started to look at the opioid class of drugs. And obviously, there are three different kinds of opioid peptides that we got very interested in. These are the endorphin, dynorphin, and enkephalin analogs. So when, whether somebody runs or when you do exercise, these endogenous opioid peptides are released. And sometimes you feel like even after a long run or after doing a tremendous amount of exercise, you don't feel pain. That's because you have a high degree of opioid peptides stimulated in your brain, and therefore they're able to suppress a lot of the pain sensations. We really are very interested in developing these opioid peptides into drugs. And so the question then came is, how can we take advantage of our nano-emulsion based delivery to enhance permeability of these opioid peptides to go into the brain? And opioid peptides obviously make a very interesting therapeutics because they're selective for various types of opioid receptors. They function in the brain. They're very, very potent. If you look at some of these examples of opioid peptides that have been tested as tool compounds, for example, the DAMME is one of those, and then DALDA or DALDA is the one we have worked with. This DALDA peptide, when it was actually administered using an invasive method in mice, shown to be as good, both DALDA and DAMME, as good as morphine in terms of its potency. However, these peptides, even though they're very small, they're only 4 or 5 amino acids long, they are not permeable across the blood-brain barrier. And therefore, anything that we can do to enhance permeability would be, a positive way to get these molecules across. The other thing with opioids, the small molecule opioids like morphine or codeine and oxycodone and other types of molecules, that they are highly abused. There is a high degree of abuse potential, and obviously there is a huge need to develop molecules that still have the potency of the opioids, but not necessarily the abuse potential of small molecule opioids. So that's another reason why we were so interested in developing these opioid peptides as therapeutic agents. But as I said, these peptides are hydrophilic, they are highly degradable, they don't cross the blood-brain barrier, and they're cleared very rapidly when they inject these into the bloodstream. So we needed to think of a multi-pronged approach to try to enhance their delivery. And so first thing we did was to take this DALDA peptide and modify it with lipids in order to enhance the dissolution in the oil phase of the emulsion. because you need to get it into the oil before you can actually package it, and it's the oil that helps us to ferry it across the blood-brain barrier. So to do that, we looked at the chemical structure, and we saw that there were two different nitrogens, one in the epsilon amino group of the lysine residue, and we modified that epsilon amino group of the lysine residue with C2 up to C8 lipid tails. In that way, we can then see whether these lipid-modified DALDA was soluble in oil, and we could make the emulsion. And we found that the C8 modified was able to dissolve very well in the oil phase of the emulsion, and also was able to retain the bioactivity as an analgesic peptide. The other thing we noticed that when we modify with these lipids, these peptides self-assemble into micelles, and that micellar structure, when it gets into, when it's present in water even, has a much greater stability against proteolytic digestion as compared to the free peptide, because the enzymes are no longer able to access the amino acids and the peptide chains as efficiently due to steric hindrance. So just the fact that you create a self-assembled structure of these labile molecules, you're able to improve their stability tremendously. But we are also interested in getting these into the oil phase. After we made the modifications of this peptide, we also attach an EIR dye to the other nitrogen which is present in this molecule. In this case, this nitrogen is critical for activity, but we attached an EIR dye in order to make these peptides so that, derivatives, so that we can then do whole body EIR imaging to show that we are able to get high levels of the peptide in the brain. And we gave these peptides that have been modified with NEIR dye, once they're put into the nano-emulsion, to our mice. These are CD1 mice. We inject the formulation by tail vein, and then we look for whole body NEIR imaging using IVIS system. And we can see that after 15 minutes after injection, tail vein injection, the emulsion form is present entirely in the systemic circulation. It remains in the blood pool for that long a time, whereas the solution form of the same peptide, the C8 modified DALDA, starts to be cleared very rapidly, and you see very low levels of the fluorescence signal coming from the solution form when injected at the same dose in these animals. And that is also seen both at 60 minutes and then more importantly also at 3 hours post-administration at 180, 3 hours post-administration, 180 minutes after administration. The solution form is almost all cleared out of the body, whereas the nano-emulsion form stays in the blood pool because of the pegylated oil droplets, which increase circulation half-life and subsequently allow us to get multiple passages into the blood pool and prevent the clearance of these peptides from the body. And the fact that we can reduce the clearance and dilution of the peptide, that is a huge and a very important reason why we want to create these types of delivery system to help us get into the bloodstream. The next series of studies that we did was to actually quantify. So we use an LC-LCMS tandem mass spectrometry method to characterize the native peptide. So in this case, C8 modified DALDA, to see how much of it is in the blood when it's given both in the solution form, which is an aqueous solution given by tail vein injection, intravenous injection, and or nano-emulsion form. So NE stands for nano-emulsion, and solution obviously is the blue circles. And you can see that the solution clears a lot faster from the body, from these mice, than the nano-emulsion. The nano-emulsion remains much more stably circulating for a long time. And when you do the pharmacokinetic parameters after you've been administered, and you can quantify, and you can see there's about a 5.5-fold higher plasma exposure and 5.5-fold lower clearance from the nano-emulsion. So the ability to keep these nano-emulsion in the blood pool, in our opinion, is a critical factor in order to enhance their permeability across the blood-brain barrier. The fact that you have very rapid clearance from the body, you never get the chance of having these molecules cross. We also then looked into the brain and see that the nano-emulsion actually helps us to get these peptides. This is the intact peptide that's basically quantified using this LC-LC-MS method, and we can see that the nano-emulsion gets us more of the peptide into the brain. And again, using brain pharmacokinetic, we see a threefold increase in the AUC in the brain with the nano-emulsion compared to the solution form. We also wanted to look at curcumin effect to see that whatever we saw in vitro with that blood-brain barrier model could be recapitulated in an in vivo model. And what we found, interestingly, is that when curcumin is either given before, so 3 hours before the nano-emulsion administration, 24 hours before the nano-emulsion administration, or given together, we don't see as much effect on brain penetration, but we see a huge effect of curcumin in terms of inhibiting the metabolism in the liver. So when you have a solution form of the drug, if you pre-treat the animals with curcumin, you can inhibit the metabolism very efficiently. Next was to test for efficacy, and we used two different approaches. We have an acute pain model that we developed, which is a behavioral model where the mice, these are naive mice, we can basically use the tail flick as a way to see the response to pain therapy. If the animal's tail is put in slightly warm water, they will flick it out very rapidly, and you can measure the time, the latent time, how long it takes for them to flick that tail out. If the animals are treated with an analgesic drug, they keep that tail a little bit longer in this warm water. So this hot water or warm water tail withdrawal assay is a very rapid and easy and straightforward way of characterizing analgesic compound. The typical time it takes for control, where the animals are not treated and they can sense the pain, is about less than 1 second. They will rapidly withdraw their tail within less than 1 second. So we can measure these time latent period that animals the tail remain, basically, and see whether that withdrawal time is extended when you have analgesic treatment. And so for this test, we use the CD1 mice. Our dose of the peptide was 3 milligram per kilogram, and we compared the nano-emulsion form of C8 modified DALDA with the solution form of C8 modified DALDA, and we also used just the native DALDA itself without the lipid modification as a control to make sure that we have not seen any change in the activity with the C8 modification. So you can see that both the native and the solution form of C8 modified DALDA, the native DALDA in solution, which is aqueous solution, and the C8 modified DALDA in aqueous solution, show this latency to about extending it about 3 to 4-fold, and it starts to appear very much within the first 30 minutes and then continues to up to about 90 minutes, and then the effect starts to wear off, so that after about 3 hours, 180 minutes, you go back to baseline. With the nano-emulsion form of C8, we see a delayed response. We see a much slower response, and the peak time then is moved to about 120 minutes, 2 hours after administration. And then we also see this extension of activity that's prolonged, and that's consistent with our blood pharmacokinetic data, because we see this long circulation time with the nano-emulsion and not necessarily getting cleared as rapidly as with solution form. And so we are able to extend the duration of activity, of analgesic activity, tremendously with the nano-emulsion form. We also looked into a chronic model of pain, in this case working with Professor Craig Ferris, who is the director of our Center for Translational Neuroimaging here at Northeastern. He has a model of capsaicin-induced pain, which is characterized using functional MRI. And functional MRI allows us to look at blood flow into the brain due to different stimuli. And whenever there is a pain sensation and the brain basically is sensing that there is pain sensation in the body, it will try to increase blood flow to certain regions of the brain. And that increase in blood flow to that region is monitored using functional MRI, and Craig and his team at the CTNI use a 7 Tesla MRI instrument to map out different regions of the brain that basically respond to the stimuli, and subsequently our approach was to, can we affect those stimuli that are induced by capsaicin with our nano-emulsion formulation of DALDA. And so we use positive bold approach to characterize with a functional MRI to characterize the effect of the peptide. In the first slide here, we again injected our peptide at 3 milligram per kilogram dose IV to these CD1 mice who were treated with capsaicin and looked at 7 different regions. We looked at the multiple regions, but the ones that are characterized here are the ones that actually did show significant differences when you are treated with the peptide as compared to control. So what you see at the bottom are animals that have been just treated with vehicle as a negative control, no capsaicin, and then on the far right are the capsaicin-treated animals, and in the middle there, where you see significant difference at less than 0.05, are animals who have been treated with the peptide before treatment with capsaicin, and this pre-treatment with peptide actually prevented the response to capsaicin tremendously in those regions of the brain. And this effect lasted for 30 minutes here, and then also for 90 minutes post-administration. So we see this long-term duration effect, which is consistently what we have seen with the PK data, as well as with our tail withdrawal assay, and now, more importantly, using this fMRI, directly looking into the brain regions to see how the activity is affected by the peptide administration. So overall, we've been very impressed by the fact that we have the opportunity to use a nano-emulsion form made with fish oil, which is rich in omega-3 fatty acids, help us to ferry these molecules across the brain from the blood when injected intravenously, and opportunity to work with very difficult molecules like peptides, which have tremendous potential as therapeutics in getting them across the blood-brain barrier. But again, our strategy is this multifold, that you don't just want to develop these into formulation, but more importantly also think about ways that we can create a modified version of this peptide to enhance stability and enhance circulation half-life in the body, and subsequently create an opportunity for these peptides to cross the blood-brain barrier. The second example that I'll talk about is a work that we are doing with Parkinson's, and specifically asking the question about neuroinflammation. So Parkinson's and Alzheimer's and a lot of neurodegenerative diseases are developed. These are diseases that affect the elderly, and they are some of the most challenging diseases of our times, because there is no diagnostic methods right now that is available to allow us to detect these diseases early. Treatments are also very symptomatic. They don't really address the underlying pathophysiology of the disease. And so one of the common denominator with a lot of the different neurodegenerative diseases is this neuroinflammation. And what you see in this slide is that in Parkinson's or Alzheimer's, once the insult starts to build up, the inflammatory molecules are secreted, and these brain resident macrophages, or what I call microglia, starts to stimulate production of various chemokines and cytokines that starts to create tissue destruction. So our approach to much more generally thinking, how can we approach not just Parkinson's or not just Alzheimer's, but think about a way that we can control neuroinflammation in the brain. And to do that, again, we are thinking about anti-inflammatory therapies, specifically that could be delivered into the brain and help us in treatment of these effects, the neuroinflammatory response that occurs because of Parkinson's, Alzheimer's, and other neurodegenerative diseases. So our target here is to think about getting these molecules into the brain. But in this approach, I'll talk about our second method, which is to get drugs from the nose to the brain. This work is been done by Sunita Yadav, again, another PhD student in my lab who graduated a year ago. And she was interested in looking at intranasal delivery as a mechanism to get peptides and small interfering RNA into the brain, especially to treat neuroinflammation. And as I mentioned before, in the case of intranasal delivery, it bypasses the blood-brain barrier. There are different theories as far as what mechanism of transport is most appropriate. People suggest it may be involved olfactory bulb and olfactory neurons. There's also the trigeminal pathway, and then also there is some understanding that it may be a paracellular pathway that allows us. But what we find really interesting about intranasal delivery is that for labile molecules, large molecules, it actually allows us to get fairly decent levels of the bioavailability in the brain, And especially with potent molecules like peptides that I'll talk about in siRNA, you can have substantial amount of the molecules delivered into the brain, and more importantly, have a therapeutic response from these molecules, and also helps to prevent systemic side effects. And I'll talk a little bit about that in the context of neuroinflammation in the example of molecule we have used. So our approach has been to use cyclosporine and TNF silencing siRNA intranasal delivery into the brain for treatment of neuroinflammation. And cyclosporine is a well-known peptide. It's a hydrophobic peptide. It's a 10 amino acids, and it's cyclic, it's cyclized, and it doesn't require any modification because cyclosporine dissolves very well in these omega-3 flaxseed or fish oil. And also in this case, we make these emulsions for delivery of these intranasal formulations by using sterolamine to enhance the residence time in the nose and subsequently allow us to get uptake and get into the brain. The TNF silencing siRNA was pre-complex with DOTAP, and that allowed us to get it into the emulsion as well. And subsequently, the emulsions were formulated both with the ultrasound method as well as the use of the microfluidizer. So what Sunita did was to encapsulate these two different molecules and then test to see if she could actually see an anti-inflammatory response. So our first test was to take macrophage cell lines, these kind of J77 cell lines, which are murine macrophages, and we stimulate them with LPS, which is a toll-like receptor agonist. And the LPS stimulation produces very, very strong inflammatory response. We see upregulation of TNF, IL-6, IL-1, INOS, many other pro-inflammatory cytokine and chemokine. And so by using cyclosporine, in this case, delivered both in solution again and in the nano-emulsion, we wanted to see if we can suppress the pro-inflammatory response in macrophages. And that will give us some understanding of what dose is needed, how long is the duration of activity, and some other parameters that we can then take to the in vivo studies. So clearly you can see from these graphs that we can, with the nano-emulsion form, we are able to suppress the pro-inflammatory cytokines, especially TNF-alpha, IL-6, IL-1, and INOS, and using the nano-emulsion form when it's administered into these macrophages. We then went to test whether we could actually get the peptides, in this case cyclosporine, in the brain and compare the brain distribution when the cyclosporine is administered intranasally and compare that to the IV formulation. We wanted to see which one is better. Should we continue to work with IV formulation or should we use intranasal? And you find that intranasally delivered peptides, so in this case cyclosporine is also a substrate for P-glycoprotein, and you see that intranasally delivered peptide actually gives us much higher bioavailability in the brain. But also we find that the bioavailability is preferentially in the midbrain region, the regions of the brain that are critical for some of these inflammatory response, especially like the substantia nigra area where the dopamine secreting neurons are present that are destroyed in Parkinson's disease. So with this data, we were excited to then move forward and see how cyclosporine could become a therapeutic for treatment of neuroinflammation. As you know, cyclosporine is a drug right now used for prevention of rejection, organ rejection, but it's also a very important drug as an anti-inflammatory molecule, and subsequently there's a lot of interest in using cyclosporine for anti-inflammatory, but the problem is that it is very toxic, especially to the liver and kidneys when it's administered in the systemic circulation. So what you see in this slide is that when we give intranasally, we actually avoid peripheral distribution because the rest of the dose goes into the GI tract and then gets cleared from the GI tract, doesn't get absorbed. And therefore, you have a very efficient way of getting the molecule into the brain and preventing systemic distribution, which means that you can actually avoid a lot of the side effects with these potent and toxic molecules. We then went on to develop a model of neuroinflammation. This is a surgical model using Sprague-Dawley rats. The LPS in this case is administered using a catheter into the brain, into the substantia nigra region of the brain, which is what I mentioned is critical in Parkinson's disease. And once the LPS is administered to that area of the brain, it generates the inflammatory signals, and we can monitor. So we looked at the effect of LPS dose on TNF and INOS levels in the brain, as well as how long that effect persists. So we find that the ideal LPS dose is around 2 milligram per kilogram given to these 2 microgram or 4 microgram, and then the duration remains around 6 hours post-administration. And so we chose 2 microgram dose of LPS per rat, and then developed the neuro-inflammation model, and then took these animals and tested our cyclosporine therapy to see how we can suppress the inflammatory response. And so here you see that when you have these animals treated with cyclosporine, there is a significant reduction in the TNF levels in the brain. You see the levels after LPS administration skyrocket up to almost 20,000-fold above baseline, and then but you see a significant reduction after treating with cyclosporine. When you see the same effect with IL-2, we didn't see as much effect of INOS, and that may be just the kinetics that we're looking at. In this case, we're looking at 24 hours post-administration. Maybe INOS changes occur either earlier or later time points, so we have to look at other time points as well. And then IL-6 also, we didn't see a strong response with cyclosporine in vivo, but we did see effects of both the INOS and IL-6 when we gave TNF silencing siRNA. So that's another formulation, again, delivered with these nano-emulsion intranasally to try to inhibit neuroinflammation, the TNF upregulation with neuroinflammation. And we have done those studies as well. We're just writing those manuscripts and submitting it for publication. And then lastly, when we looked at histology, what you see here are the microglia. So you have on the top left-hand side, no treatment, and just giving LPS on one side of the brain. As I mentioned, we are putting it with a catheter. So you see that that side of the brain, the brown stain is activated microglia. Basically, when LPS is given, these microglia, these brain-resident macrophages, converge to that area and become activated. And the shape of these microglia is an indicator of activation. When we treat with solution, we see some changes in the levels of microglia, but not significant compared to the untreated side. Untreated side is always our control. But the treatment with cyclosporine clearly shows a decrease in the levels of these activated microglia compared, and we can quantify. So these are just 3 different sections from these animals, but we can quantify it using 10 different sections and collect the percent intensity and compare it to the contralateral side, which is not injected with LPS as a control, and that percent change is significantly different with the nano-emulsion treatment with cyclosporine compared to any of the other formulations, both the solution formulation of cyclosporine and more importantly, the control LPS treated. So with this, let me just summarize what I've covered so far. Clearly, there is a tremendous need in development of therapeutics for brain diseases, especially chronic brain diseases such as pain and neuroinflammation. And what we want to use are the biological approach to therapeutics that actually modify the disease rather than just treat the symptoms. And obviously the challenge, as many of us know in brain delivery, is the fact that it's very difficult to get molecules into the brain through the systemic side due to not only the blood-brain barrier, but also the issue of systemic dilution and clearance. And our approach has been to try to create non-invasive mechanisms. Although there are invasive and non-invasive mechanisms of delivery into the brain, the non-invasives make a lot of sense because you can't drill a hole in the skull and try to get drugs through a catheter or use mannitol to enhance penetration into the brain because these are very transient and they have a lot of toxicity associated with them. So the idea of using any of these invasive approach on a clinical as a routine clinical approach for treatment is just not feasible. We have to find solutions that are non-invasive, that are patient compliant, and ideally allow us to then develop these therapeutics so that they can help in treating patients. So I talked about our use of these nano-emulsion formulation for encapsulating the analgesic peptide DALDA, the opioid peptide. It's a mu opioid peptide, and showing both the delivery efficiency as well as efficacy in 2 different models, the hot tail withdrawal assay as well as the capsaicin-induced model and fMRI studies. And then I also showed the intranasal approach for delivery of cyclosporine, and we also done work with intranasal delivery of TNF silencing siRNA to enhance in treatment of neuroinflammation. One of the key approach thinking that we have in our lab is really to make sure that these are safe delivery vehicles that we design. So we use edible oils, we use surfactants that either come from food or from natural compounds. We use safe materials, scalable methods to develop the nano-emulsion. As Steve mentioned, the microfluidizer allows you to scale these formulations from small volumes such as 15 ml all the way to liter quantities and GMP manufacturing capability. And those are really needed for translating all these different ideas from experimental approaches to make them clinically viable therapeutic strategy. So again, thank you for your patience and being with us this morning. I'm happy to answer questions if we have some time. I'm also accessible, as Steve mentioned. I'm welcome anything that you have by email, and certainly I'll be happy to respond to any questions that come up. So thank you.
Thank you very much, Dr. Amiji. That was an excellent webinar. I appreciate your time, and it's been a very interesting development that you guys have done both with for both techniques. So we have a couple of questions that have come in through the website. The first one I'm going to read it talks about, what about structure and polymorphism of drugs in the nano-emulsions? Are they characterized, example, by solid state NMR, and is this an issue?
So thank you, Steve. Certainly, I think one of the good things about biological therapeutics like peptides especially is that the structures, as long as the amino acid chains are known and we can characterize the peptides, in this case, if it's the DALDA, it's a 4 amino acid peptide, cyclosporine is a 10 amino acid peptide. So we do characterize our precursors very well, and we want to make sure that they are they are active as free agents, as free drugs. So in this case, the polymorphism is not really that much of an issue with peptides as it is with small molecules. If we had crystals, for instance, it would be a huge problem because with nanocrystals, for instance, that's a big problem because you grind these, you crystallize the small crystals, then dissolve, and they're in continuous equilibrium with soluble agents, and then they reprecipitate, and it's the reprecipitated form that tends to be polymorphic, and the changes that occur in the reprecipitated form is what creates a lot of problems. Could have very different bioavailability and different dissolution profile. But the good thing about nano-emulsion is that as long as you can keep the drug in solution in the oil phase and keep it inside so it doesn't partition out into the aqueous phase, and that's why we made a lipid modified DALDA, because we wanted to keep it inside the oil phase and remain there in the oil phase and doesn't partition out into the aqueous phase to try to, and then that protection that comes from the oil phase is critical in order to improve its stability as well as prevent it from clearing out of the body.
Thank you very much, Dr. Amiji. Here's another question. The question came in, it's what type of post-production purification do you employ?
So yeah, this is a very good question, especially for both intranasal and for IV administration. So the good thing about nano-emulsion, and I think part of the whole issue with preparing nano formulations of drugs is that they are sterile filterable. You can filter them using 0.22 micron. Because the size, as I mentioned with our DALDA nano-emulsion, the size was around 100 nanometers. Same thing is true for the cyclosporine nano-emulsion. The size of the droplets is around 100 nanometer. So they're easily sterile filtered through 0.22 micron, and that allows us to create a sterile formulation at the end. But the other key component to this are anything that activates the immune system. So obviously making sure that your raw materials that you're starting with are endotoxin free. And we go through a lot of, in some cases, it's a tedious process to remove endotoxin if you have endotoxin in your raw material. But ideally, it would be good to just start with materials that are endotoxin free in order to make sure that your product remains endotoxin free as well. And then containers and vessels that are used in packaging or in preparation of these systems also remain to be fairly well cleaned to ensure that you are not contributing to anything inside the formulation that ultimately will have some reaction. One of the key things that I think most people miss in nanoformulation design are the immunological consequences. So endotoxins, anything that can affect the immune system, such as complement activation, it becomes really critical. So, some of the in vitro tests can be done. Like in endotoxin tests are very easy to do. There is a test called LAL, uses crab blood, and you can characterize whether your product has endotoxins or not. So that would be really easy test to do, but also help you to make sure that your product doesn't really have any contaminants when you're administering to cells and, more importantly, to animals.
Thank you. That's very clear. We have another question that just came in. It is, what is the stability of nano-emulsions, and under what conditions did you analyze the stability?
So, good question again. There are several ways to characterize stability of nano-emulsion. I mentioned a couple in the discussion related to the DALDA peptide. So the most common is if you're just testing stability in vitro, size of the droplet and changes in size of the droplet, data potential values over time is a very easy thing to do. You just take a small volume at different time points under different conditions of storage, and then measure the size and chart changes over time. And ideally, you shouldn't see more than very small change, daily variation in the size and charge. And so we do this in our lab. Again, this is at the lab scale, not necessarily at the production scale, for up to 6 months. We want to make sure that our products are stable, both at room temperature and also at 4 degrees. So we do it at 2 different temperature conditions to make sure that in some cases, obviously, there will be longer stability in the refrigerator compared to room temperature. But we also look at the drug encapsulated inside, how long, so we have an HPLC assay for these peptides, and we'll look at the changes in the concentration of the peptide over time. That's an indicator of how much drug degradation is occurring, if any. The other very important is just visual observation of the emulsion. If you have a poorly prepared emulsion, if the surfactant concentrations are not optimized, what you find is that the oil and water separates. So that's an example of a bad salad dressing, where when you mix the oil and vinegar or oil and other components, you're not they separate out very quickly. And so if you see that the oil layer is on top and the water-rich layer starts to be on the bottom, that means that you have an unstable system. Now, there's another phenomena which is confused with phase separation. It's called creaming effect of emulsion. In this case, you can actually have the emulsion separate, but the oil-rich phase then floats and the water-rich phase then settles. But you can stir that, and it's reversible. So that's not necessarily a stability question. That's just the fact that you have these 2 layers separating out. But you can actually shake the emulsion. It will go back. And again, if you measure the particle size, it will be exactly the same as what you found before. What the problem would be if you get complete phase separation, meaning that you actually see the oil, in this case, in our case, like fish oil, you see this yellow band on top, or flaxseed oil, you see a yellow band on top. That clearly tells you that there is phase separation, and that's not reversible.
Okay, great. Thank you. So I think there are 2 more questions that just came in, so we'll go through these real quick and then we'll wrap it up. The first one is, were there any differences in the stability of the nano-emulsions that were prepared by the sonicator versus the microfluidizer?
Yeah, again, great question. Yes, there were actually. So the sonicator, it's a great tool for lab scale preparation. We use this probe sonicator. There's a company in Connecticut that, and we list that name of the company in any of our publications that use with nano-emulsions. and specifically create a coarse emulsion, and then you pass it through this probe sonicator, and you allow you to get the nano-sized. But as you saw from that table, the droplet size varies with the probe sonicator. It's one of those things that you really have to optimize the condition to get it right. So we get around 200 nanometers, sometimes 250 nanometer droplet size with the probe sonicator. Whereas with the microfluidizer, it's very reproducible. We get 100 or less each and every time, as long as we use the same conditions. So in our system, 16,000 to 20,000 psi, and we'd run it at least 3 to 4 cycles, and again make the coarse emulsion and then just pass it through the microfluidizer. Stability-wise, clearly the sonicator sometimes gives us some stability issues. Droplet size starts to change. We've not seen any problems with the microfluidizer as yet. Again, for our test, again, it's a 6 months test, both at room temperature and refrigerator temperature. We have not seen size changes with the microfluidizer. So the key to the formulation is the incorporation of surfactants and making sure that you optimize the surfactants.
Thank you. All right. One final question. You mentioned that the cyclosporin had adverse health effects. What differences are there, if any, between side effects if you deliver the material intranasally versus the other methods of delivery?
So, one of the things that we think about a lot is, how do you get these molecules into the right place in the body? But then there's also this question that is really important in terms of therapeutics, is what happens when they go to the wrong place? So if you deliver cyclosporin, right now obviously cyclosporin emulsions are given orally for organ rejection. When somebody gets kidney transplant, for example, they'll be on cyclosporin therapy. They take these products such as Neoral, which is a microemulsion system of cyclosporin, and it's given orally. That product gets absorbed from the gastrointestinal tract, gets into the liver, and subsequently from liver it can go into the bloodstream. And that's how the product works, is by suppressing the immune system so that the transplanted organ remains viable in the body. But the challenge with the cyclosporin is that it is extremely toxic to kidney and liver when it's given in that way. And we see the same effect when we injected the cyclosporin nano-emulsion by tail vein in these are rat studies, and we showed that when it gets into the liver and when it gets into the kidneys upon systemic administration, there is this toxicity issue of cyclosporin because it distributes all over the body. What happens with intranasal is that you focus now only in the brain. The challenge with intranasal is the bioavailability is low, so only about 1 to 2% of your dose gets into the brain, which limits, if you don't work with potent molecules, it wouldn't be efficacious. But if you have a potent molecule like cyclosporin or like TNF silencing siRNA, it is therapeutically effective. The rest of it goes directly into the GI tract and then just gets cleared out of the body. So our nano-emulsion, unlike the microemulsions that Neural makes, are not absorbed. And so we can clear it out of the GI tract. And so you don't get any systemic exposure, at least not to the level that you get when you give product by IV, by tail vein.
Thank you very much, Dr. Amiji. We truly appreciate your willingness to speak to our customers and to our prospects. It's been a very interesting webinar, and if anybody has any questions going forward, please send them either to me or to Dr. Amiji. I'll send you his contact information after this call. So again, thank you very much, Dr. Amiji, and thanks to everybody who attended.